Assalamu alaikum wa rahmatullahi wa barakatuh. A very good morning to all the students. Dr. Rakesh Kumar Sharma is a very, very old friend, a mentor, a guide to me. And Dr. Jalal, Dr. Naseef. I think as a teacher is very happy when his class is full. I'm really very happy that you are attending in good strength and you came all the way from different parts of Kerala, different parts of India to be a part of Profcon, which is a very, very, I think, exciting event which is going on. Today, I will just discuss what is happening in the world in the field of science, especially medical science, because my area of research is drug delivery, drug targeting, nanomedicine, nanoparticles. For those students who are not have knowledge about drug delivery or drug targeting or pharmacy, when we discover a new molecule is in the form of a chemical, which is known as API, active pharmaceutical ingredient. So you cannot give that molecule directly in pure form to the patient, because that molecule has its own physical and chemical properties, its own solubility, its own absorption, and if you give it in the pure form, maybe it's not be able to perform its action at the right place at the right time. So as a pharmacy person or pharmaceutical scientist, we have to study the physical chemical properties of that drug and make it in such a form, design it at such a platform that it should be able to perform its action and cause minimum damage to the human body, to the patient. So if you see in the global R&D, it's working. If you see in India, why India we are talking about research and why? If you see in the US, the R&D spending is very high, while in India is less than 10%, while in abroad the R&D spending is more than 30%. And they are investing very heavily in research and development. Companies of America and Japan, they all have about 250 R&D facilities in USA. Then you can imagine if they have 250 R&D centers, how much amount of research they are daily doing. More than 100 R&D centers they are setting up in India. So now, currently the emphasis is now shifting to external technology acquisition to complement the internal efforts. What is external technology acquisition? It is basically, you must be reading in the newspapers that many companies, they are purchasing or licensing out or licensing in technologies developed by different companies, small startups. Especially in the area of software engineering, many small startups are coming up every day. And after developing a normal app or developing a software, they are simply selling it off or giving it, licensing out to bigger players, MNCs. And that is a very emerging trend. Same happening is in pharma world also, in medicine also. In biotechnology also, you must have heard about Shanta Biotech, Biocon. Daily they are developing new projects and selling those projects to bigger players so they can earn the revenues and then further work on or further invest in the field of R&D. So suitable taxation and related arrangements has to be developed in the country which can prosper the research and development efforts of the industry. Suitable appropriate legal framework has to be established, which is in India. Now we have newer IPR laws since 2005. Old IPR laws, if you see, what is IPR is intellectual property rights. It is like a property right, you have right to whatever product you are developing out of your intellect. It can be a medicine, it can be a software, it can be a artwork, it can be a article. Whatever you are developing for the first time in the world, you can apply for IPR patent, you can apply for copyright, you can apply for trademark, and the purpose is to encourage sharing of knowledge. When you are sharing your knowledge which you have developed, you are giving chance to other researchers to work on the same problem, and you encouraging further development. And in return, the government is giving you right for about 20 years that you can use your idea for commercial benefits. Company can sell the product and nobody can use it without permission. If they are using it, you can take them to court and ask for damages. Now, I will just see what is the strength in India. In CSI labs, you can see the number of scientists. We have more than 10,000 scientists who are working in CSI labs. In ICR or agriculture universities, we have more than 6,000 scientists who are working. In ICMR-based R&D institutes, we have about 700 scientists working. In Department of Biotechnology R&D institutes, we have about 700 scientists who are working there. In Defense Research Development Organization R&D institutes, we have more than 6,000 scientists working in different areas of research. In Department of Education, if you see, we have more than 5,000 scientists. In ISRO, Department of Science, we have more than 10,000 scientists working. And you can see how many researchers are working in the area of various fields. In universities, you see, you have 30,000 in colleges. Around IITs, we have 7 IITs, where we have about more than 2,500 faculty members, students, 23,000. So our advantage is our technical manpower, which is available at a much less cost as compared to European countries and American countries. And that is why foreign companies are very much interested in developing R&D centers in India. US FDA has established its biggest R&D or analytical validation center in Hyderabad, because the cost of development, the cost of manpower is much less as compared to USA. So the emerging trends is FDI or foreign direct investment. It is now permitted in India up to 100% through automatic roll. The Indian is also agreed to implement a product patent regime. Earlier, we are following only process patent regime. But now, from 2005 onwards, companies can register or patent products. And this has allowed the foreign companies to launch their products in India. If you see, now there is an increased investment by pharma companies in research and development. Many companies, they are coming up with their R&D centers to develop new products and to generate their own IPR. So R&D can be performed in India at about less than 20% cost as compared to international arena. So that is why they are investing in India, because they are getting very technically qualified manpower and getting the manpower at much less cost. Indian companies are pursuing contract manufacturing for supply of bulk drugs, intermediates for many multinational companies. Companies like Mankind, Pharma, they are all doing the jobs for MNCs. There are many companies where they are manufacturing the products of MNCs. So because of low cost production, India has become a base for outsourcing bulk as well as outsourcing formulation, outsourcing chemicals, biotechnology based products. They are all coming to India. So it is a hub for testing and development of new drugs. Why? Because pharma industry today takes about $300 to $700 million, and it's up to 10 to 15 years to introduce a new drug in the market. 15 years is a very long period of time. And after investing 15 to 20 years to develop a new product, a new drug, you are not sure that what will happen to that drug. You must have heard stories about coccyx. Celecoxib when it was launched was supposed to be a miracle drug. But even after launching into the market, they showed signs of various types of toxicities and side effects, and they have to withdraw. So you can imagine, after investing 20 years and about $700 million US, they have to withdraw the product. So the chances are very rare. So we have a large pool of well-qualified and skilled manpower. We have availability of large number of patients with all forms of diseases. Because of the population, we have good number of patients available for their clinical trials, for their clinical evaluation. We also have patients who have not received any other medical treatment. Because of non-availability of medical facilities or because of poor nature of the population, we have patients who are never given a drug. So this gives a chance to multinationals to evaluate their drugs for the first time in humans. So another area where we have to focus is database search. As Professor Sharma was saying, now you have your classroom on your mobiles, on your laptops. Similarly, you can search your library on your mobiles. You have various types of databases, chemical databases, SciBases, Science Direct, where you can do your complete theoretical search and decide about your future direction in your practical, in your product development or in your software development. You can simply go through those databases and you will, on a click of search, if you can identify your good keywords, if you are very good in identifying keywords, then you can make a thorough research and evaluate what type of work is already been done. So you can work beyond that. You're not doing a repetitive work. You can just go through it. Plagiarism is another hot area where you have to be very, very careful that what you are doing today, it should not be, you should not be infringing someone else's work. So on the basis of these data searches, you can find out whether the work which you are thinking about is not infringing someone else's creation. You can see you have Science Sci-Fi Finder, STN, Science IP, CAS databases. They're all available, although they are available at a cost. But in universities and educational institutes, they are provided at much less cost through UGC. You can see the patenting of new chemical research, how the countries are following. You can see the graph. This is how the percentage of new compounds they are added to CAS registry, Chemical Abstracts Registry. And you can see patent informatics is a new area where you have to work in the area of IPR. You can find out what is happening in the world and then you can advise your company that this area is naive where nobody has worked and company can gain by inventing or developing new technologies in that area and applying for patents. And patent is wealth. If you have patent, nobody can infringe it, nobody can copy it, and you can gain commercial benefits by marketing it, by selling it to different companies, to population, and to in different marketplaces. So all these such technologies for drug discovery, they are available. Now, if you look at the drug discovery process, it's a very exhaustive process. You have to do a target selection. You have to do complete molecular studies, how it is going to happen in the molecular studies. Then you have to go through the animal studies, either in silico or in vitro or in vivo, doing personally on the animals if they are available to you. And then it leads to your target selection. And from there you can design your lab synthesis process and you perform your mechanism studies there. And then you have gone into the development where you can do preclinical studies, your safety studies, your efficacy studies, and then you start your basic clinical trials, phase 0, phase 1, phase 2, phase 3, and phase 4. So all these are required for. So when you are doing for a target selection, first you have to decide in which area you are selecting a target. What is the unmet medical need? Many companies are working in the area of cancer because we don't have a effective or treatment for cancer. Although many anti-cancer remedies are available in the market, but you have such a huge mutation and such a huge variety of different types of mutations happening in the human body that one treatment is not sufficient. So similarly, you have to select an area. And then after selecting the area, you will be performing in vitro studies, in vivo animal model, pharmacodynamic, pharmacokinetic studies. You have to identify a lead compound. And out of that lead compound, then you have to perform thorough safety studies as per regulatory guidelines. Which regulation is controlling the new drug development? You are going through Schedule Y of Drug Control Act or Schedule M. So mostly in the initial studies for clinical studies, you are following Schedule Y, which is define the ways and means how to do the research in this area. Then in the development, you have preclinical development, where we are doing all the studies in animals or in software models or on cell lines. So after from cell lines, you are transferring to animals from one animal to another animal species, rats, mice, rabbits, depending upon where you have an effective model of your target disease. If you are working in tumors, so which tumor can be developed in rat, which tumor can be developed in mice or rabbit, that you have to decide depending upon your data search or literature search. If model is not available, you have to yourself develop the animal model for your particular disease, which can mimic the same condition as in human being. Then, after doing these studies, your scale up, synthetic development, and then pre-formulation and formulation studies. You will go for your marketing strategy and your clinical trial, and then approval for 2 regulatory bodies. You will be filing for IND after doing your phase 1, phase 2, and phase 3 studies. And then when you file IND and you file all the data to the US FDA or Indian FDA, then you will get the final approval. So this is how you are submitting and getting the approval for a new drug research. See, these are it is a multidisciplinary area. It is not only about pharma or about chemical industry. You can see how many areas which are converging in the development of a new drug. See all the areas. You can see combinatorial chemistry is there, synthetic chemistry is there, patent laws are there, physiology, biochemistry, pharmacology, immunology, your drug metabolism, and your enzymology, your physical chemistry, behavioral science, pathology, pharmacology, safety assessment, metabolism, all is compiled into one, joined together to give you a successful drug. All these you can see. Now, coming back to, I have listed some of the examples, some success examples, which are some of the platforms where we can work for designing or for giving a drug delivery system. Either they can be colloidal carriers. Colloidal carriers are basically, they are small vesicles which are in the micron size range, and they are made up of various colloidal materials. And these vesicles, they can encapsulate the drug. They can be used for targeting. Targeting means when we take a simple tablet by oral route, it is a immediate release tablet, like you take Disprin, or you take paracetamol tablet, crocin tablet. So they are not targeted. They are simply immediate release products. When you ingest them, they go to the stomach, release the drug is absorbed through GIT, and through blood, it reaches the site where you have pain or inflammation in the body. In targeting, what we are doing, we are using strong drugs like anti-cancer drugs and targeting them to a specific tumor. Why? Because we want to have maximum effect at the site of infection, and then we want to minimize the toxic effect to other parts of the body, other cells of the body. That is why we are doing targeting. So by colloidal carriers or cellular carriers, you can achieve this targeting. You have different types of these systems. You have micelles, reverse micelles, mixed micelles, polymeric micelles, liquid crystals, liver proteins. You have polymer-based systems like signal-sensitive systems, mucoadhesive systems, biodegradable, bioreadable, all these terms. Then you have macromolecules based, where you are using proteins and peptides as a carrier. You can see the different types of vessels, micelles, dendritic polymers, nanospheres, and you can see liposomes. Now, I just will give you some examples. These are the commercially available products, which is Doxil, which is a very popular drug in the form of, available in the form of lipidic carrier. Then you have daunorubicin, amphotericin B, tretinoin, which is used already in phase 3 studies, nystatin in the form of cream, amikacin, prostaglandin. Yeah, they are all success stories. So from these success stories, you can see that even you can design these fancy molecules into a product. You can see this is already available in the market. Doxorubicin, liposomes injection, ambisome, which is amphotericin B liposome. Amphotericin B is highly toxic. Its toxicity is much reduced when it is given in this modern form, in the injectable form. You have liposomal doxorubicin, which is available in the market in the name of Calyx and Myoset. Then you have neosomes, which are surfactant-based molecules. These are the 3 major types. The success story is Timolate, which is a nusomal preparation of timolol maleate. And there you can see pharmacosomes, virosomes, we are using various carriers, and they contain a viral envelope which is inactive, and it contains drug, and then it has its own lipoproteins on the surface by which we can target to a specific cell or group of cells. These are the various types of surface, you can say, targeting ligands we can use on the surface of these virosomes. We have cytokines, we have peptides, monoclonal antibodies which can be used. You can see these are available products for influenza vaccine, which is known as Inflexel V. Then we have Apex Aluminium, which is the only virosomal adjuvant hepatitis A vaccine. Then you have, you must be hearing about nanotechnology. Nanotechnology is not a new science. Nanotechnology is already there in the nature. In our body, there are particles which are in the size range of less than 1 micron. Earlier, we don't have electron microscope. We don't have AFM, atomic force microscope. After the development of atomic force microscopes and electron microscope, now we can manipulate materials at a nano size range. That is why now nanotechnology becomes very, very important. Why nanotechnology is making so much of, you can say, sound in the market? Because by applying nanotechnology, our own material, which is having some specific properties, its properties are changing. Why? Because in the micron size range, you have the bulk property of the material. The moment you goes to the nano size range, its surface properties becomes more prominent. Surface area is increased to a maximum level. And when surface properties becomes more prominent, the material becomes highly active. Because by reducing size, you are increasing the surface area. So there are many materials in the micron size range, they have different property. In the nano size range, their properties completely changed. In the drug delivery, we are taking advantage of size for penetrating into various organs of various tissues of the body. Because these small particles, they can penetrate deep into the tissues, they can bypass various protective mechanisms of the body and attack the bacteria, or I can attack the tissue or tumors deep into the body. You can see the example of biosilicon, which is a silicon-based nanomaterial. And it is an inert material. It is patented by an Australian company, Chivida, and they are using it as a platform technology for delivering drugs. Then you have carbon molecules, fluorines, buckyballs. You heard about it. Then we have metallic nanoparticles, gold nanoparticles, which are used for in the anti-cancer therapy. You have dendrimers. They are branched macromolecules, which can be used for very, very sensitive molecules. They can encapsulate them. Then you have nanosite self-assembly liquid structures like molecules like lycopene, beta-carotene, lutein, phytosterols, coenzyme Q10, DHA, EPA, they all can be encapsulated and can be delivered in the human body. These are the marketed products, Abraxane. You can see here you have Medusa, which is a self-assembled polyamino acid nanoparticle system. It is a versatile protein carrier for the development of novel and second generation long-active native protein drugs. Insulin, then you have microspheres. You can see you have mitomycin, 5-fluorosyl microspheres, progesterone microspheres, adenomycin proteins, cisplatin, and various enzymes. Then you have magnetic microspheres. The magnetic microspheres, they are basically iron oxide microspheres which can be injected into the body, and by applying a magnetic field outside the body, you can concentrate them at a particular location, and there they can release their drug. These are the products which are already available in the market. Then you have various types of micellar products, dendrimers, multi-branch systems, where drug can be encapsulated into their branches, it can be protected, and it can be delivered also. Here are examples which I am sharing. This is one work which we have done in our lab, where we have developed product for targeting to pulmonary system. And you can see we have developed with INMAS and DRDO, where we have shown, this is the images of a human lungs. And if you see, on the left-hand side, you have a dry powder inhaler given to a patient in the micron size range. On the right-hand side, where dry powder inhaler is given the nano size range. And in the nano size range, you can see, after 6 hours, the lungs are still visible. These are the images they are taken by a gamma camera, and drug is tagged with technetium. It is a radio emitting system. When technetium is attached to the drug, it can be detected by a gamma camera, and you can see where your drug is going. And here you can see in the nano size range, more amount of drug going into the lungs, and it is retaining there for more period of time. This technology we are using for the treatment of lung carcinoma, chronic obstructive pulmonary disease, pulmonary hypertension, and asthma. This is a few examples. We have used salbutamol, it is already patented and developed into a nano DPI. When it is given to the soldiers at high altitude, their exercise capacity is increased by 30%, their pulmonary hypertension is reduced, and with a single puff, their COPD condition is highly improved. They are already using it at their high altitudes. It has gone through phase 2 and phase 3 clinical trial, and they are used now, the technology is transferred to forces, armed forces. They are using at Leh and Ladakh bases. This is the same technology they are using for delivering antidotes to the lungs. Now, in the end, I will just tell you about my university. It was Jamia Hamdard, established as a philanthropy by Hakim Abdul Hamid in 1962. He is the owner of Hamdard Waqf Labs, who is the largest manufacturer of Unani products. He donated all the earning of the company into a foundation, which is known as Hamdard National Foundation. And that foundation is maintaining various educational institutes, schools, and colleges. And one of them is Hamdard University. So this is the few images of the Hamdard University labs, buildings, our labs where we are working, systems. That's all. Thank you.